I'd like to introduce Dr. Lily Lee, our medical director, and I continue to learn from her every day. Dr. Lily Lee brings extensive experience with not just orthoclinical diagnostics, but also from Johnson & Johnson. She obtained her medical degree from Peking University in China, and then her PhD in immunology from the University of Alberta, Canada. Then she completed her postdoctoral training at the Scripps Research Institute in San Diego. And Dr. Lee is the author of more than 40 scientific articles and has filed 17 patent applications. So we're very honored to have Dr. Lily presenting this to us. I just wanted to briefly go over Ortho's history. As a brief introduction to orthoclinical diagnostics, we have a rich history of 80 years in diagnostics, from our earliest work in blood typing to the latest developments in laboratory systems. As you can see here, we have a rich history in infectious disease testing with hepatitis tests and HIV tests. We collaborated on the first test for the detection of hepatitis C. Starting with that, we continued to innovate on hepatitis C, testing and HIV tests. We then also brought to market advanced immunodiagnostics platforms, which then were, one of the first platforms for automation. And then in the past 4 years or 5 years, we have, continued to revive that legacy, and we've launched VITROS HIV combo. We brought to market our Charvet's test that is used for blood screening on the VITROS instruments. And now we brought to market the total and IgG COVID-19 antibody test. The VITROS technology is really well suited for infectious disease testing to bring you the accuracy you need. And we really believe that these COVID-19 tests are high-performing tests. And with that, I'll turn it over to Dr. Lee to tell you more about those tests.
Thank you, Dipali, for the nice introduction, and good evening, everyone. I just want to echo what Dipali said, that gave a big thank you to all of you for fighting in the front line for this terrible COVID-19 pandemic. And to us, you are the heroes, you are the inspiration. Also thank you for joining the webinar at such a late hour. And in the next 30 or 35 minutes, I'm going to give you a brief introduction of the two COVID-19 assays we recently developed at Ortho Clinical Diagnostics, their features, their clinical performance, and also their clinical utilities. So first, take a look at the disease and the virus. As for today, we're all in the midst of the global COVID-19 pandemic that affected so many countries, so many people, and also, unfortunately, so many people lost their lives because of that. The virus causing this disease and the pandemic has been identified as a novel coronavirus named SARS-CoV-2. Although COVID-2 is a novel virus, coronavirus has been with us for a long time. So as for today, there are total 7 human coronaviruses, including COVID-2. So 4 of them are called the common coronavirus that induce milder symptoms, but it's around year round. And unfortunately, the remaining 3 can induce more severe diseases, severe acute respiratory syndrome. This including the SARS virus that caused the SARS epidemics in 2003, and also the MERS-CoV virus is caused the Middle East respiratory syndrome epidemics in 2012 and 2013 timeframe, and now it's the COVID-2. On the right hand, this is a lineage relationship among these human coronaviruses. As you can see, the novel virus COVID-2 is more close to the SARS-CoV viruses. They share almost 80% protein sequence identity and 50 with the COVID-2. And the next closest relative is the MERS-CoV, and COVID-2 and the MERS-CoV share about 50% of the identity. Because of the around of these human coronaviruses, when we develop an assay to the new virus, we have to make sure that the assay has no or very minimal cross-reactivities to antibodies against the other coronaviruses, so to make the assay more specific. This is a nice picture, cartoon picture showing the structure of SARS-CoV-2. It is a single-strand RNA virus with the spike on its surface, very obvious. SARS-CoV-2 expresses 4 main structure proteins, this including the spike protein, the membrane protein, the nucleocapsid protein, and the envelope protein. The spike protein is a pretty large protein, consists of 2 subunits, S1 and S2. S1 is the distal domain of the S protein. And S protein forms as a trimer on the membrane of the virus, and this is the part of the virus bind to the receptor, the ACE2, to trigger the entry of the virus and the infection. The binding is through the so-called receptor binding domain, which is part of S1 subunit. S protein is an immunodominant protein that induces antibodies. And this table shows the common protein that is used for assay antibody serological assays. So this including the nucleocapsid protein, the S protein, S2, S1, and S2, and also the RBD domain. So when you compare among these proteins, S1 shows the least homology with the counterpart of other coronaviruses. For example, it only shows 66% homology with the SARS, while the N protein shows 90% homology. The less homology, the more unique of the protein specific to this virus, so it's less likely to induce cross-activity with other assays, with other viruses, so make the assay more specific. Also that we know most of the neutralizing antibodies attacking the surface protein of the virus, same as for SARS-CoV-2, antibody that blocks the interaction between S protein, S1, and the receptor shows the inhibition for infection in vitro. This is the neutralization activity. So antibody that can do that called neutralizing antibodies. And these antibodies also demonstrate protection from the infection in monkey models of SARS-CoV-2. In addition, that we know S protein is the target for almost all of the current vaccines that are under development. So the goal for all these vaccines is to induce the neutralizing antibody to the S protein, so prevent the entry of the virus to infect the host cells. So we believe that a assay that target S1 protein not only can be very specific, but also can detect functional antibodies. So the antibody that shows biological function to neutralize the virus. So let's take a look at the immune response after SARS infection, SARS-CoV-2 infection. This is a very nice illustration of the kinetics of different biomarkers relative to symptom onset after infection. And this is a very representative one based on many publications. And this picture was taken from a recent review article published in JAMA. So as you can see that viral RNA can be detected shortly after the symptom onset, and within 1 week, the RNA peaked, and later on, it lasted for about 3 weeks and subsequently become undetectable after 4 or 5 weeks. Seroconversion, that's the start to producing antibody, really starts in week 2 after symptom onset. And about 50% of the individual become antibody positive in week 2 or toward the end of week 2. And by week 3 and week 4, almost all patients who have been infected have antibody. The uniqueness about the antibody production after COVID-2 infection is that different isotype of antibody, IgM, IgG, IgA, they come very close to together, and rather than in distinct waves like in other viral infection. So that gives quite a bit of the heterogeneity of antibody production at the individual level. So if we look at the early seroconversion stage, the level of antibodies in the sample is quite heterogeneous. So it depends on the time of seroconversion. Sometimes IgG even come earlier than IgM, or IgA come earlier than IgG, IgM, and also the type of antibody and the level of antibodies, which oftentimes linked to the disease severity. So the more severe the patient, the higher antibody normally is produced. And probably it also depends on whether the patient is symptoms or or asymptomatic. So because of the heterogeneity and the kinetics of different isotype of antibody seroconversion, so the most sensitive assay to detect an early seroconversion really is the total antibody assay that detects all type of antibodies. And we know based on the kinetics of these biomarkers, the molecular test is the primary tool for the diagnosis of acute infection. However, as you see that after 4 to 5 weeks, the RNA in the nasal swab become reduced and even non-detectable. So this is the time where the antibody come up. So antibody or serological tests can be used as aid in the diagnosis for SARS-CoV-2 infection in combination with the PCR, especially those antibody tests that with the high sensitivity to detect the early seroconversion. So let's look into the 2 assays that recently developed at Ortho Clinical Diagnostics. The first one is the total assay. It is a quantitative assay that measures all antibodies, including IgG, IgA, and IgM. And it can be used on serum samples and EDTA plasma samples. The claim we have for the intended use is to, the test is an aid in the diagnosis of patients with suspected SARS-CoV-2 infection in conjunction with the clinical presentation and the result of other laboratory tests. And we particularly say that the test result alone cannot be used as a sole basis for diagnosis because RNA still come earlier than antibody. So on the this is the structure of the test. The test is using the VITROS microwell technology. The well is coated with a biotinylated recombinant S1 antigen S1 subunit of the S protein. And this will capture all the antibodies in the sample that can bind to S1 protein, and then the signals detected by HRP labeled S1 antigen again. So as you can see from this diagram, any antibody that can bind S1 antigen, irregardless of their isotype, IgG, IgA, or IgM, can be detected by this assay. And also this so-called sandwich structure is the type of design really provides high analytical sensitivities and specificity, so that translated to also high clinical sensitivity and specificity we will review a bit later. So again, like we just discussed, because of the kinetic heterogeneity of seroconversion among different isotypes of antibody, we believe that a total assay provides the highest clinical sensitivity in compared to just the IgM only or IgG only test. So traditionally, we think IgM is more sensitive because IgM come earlier. However, that's not always the case for SARS-CoV-2 infection or in COVID-19 patient. So a total assay, no matter which isotype come first, you will capture that and make this assay more sensitive. Because we use S1 protein as the capture and detection protein, this assay will also detect the neutralizing antibodies that can bind to S1 and block the interaction between S1 and the receptor. So again, it's not only detect the biomarker, but also detect the functional antibodies. And this assay is not sensitive to biotin interferon. So the sensitivity of this assay has been evaluated using 86 samples. These are the PCR positive sample for COVID-2 infections from 86 individual patients. So each sample is a patient. I want to mention that oftentimes if you look into some package insert of some assays that they use serial collected sample, which means less patients, but the serial collection of from one patient. That definitely is acceptable, but it shows less power than samples collected from individual patients, because if the serial collection, once you're serial converted, all the samples past that date become positive. So definitely samples coming from individual patients shows more power. So our assay always evaluated by using samples from individual patients. Because RNA always come earlier than antibodies, so not all the PCR positive samples will contain antibodies. So it is important to characterize the sample, particularly for the collection time in relationship of symptom onset. So among those 86 samples, 20 were collected within 8 days of symptom onset, and 49 samples collected after 8 days of symptom onset. Unfortunately, for 17 samples, we do not know the collection date, so we cannot analyze according to the symptom onset. So this assay, again, is a qualitative assay, express the results as signal to cutoff ratio. So if the ratio is lower than 1, that's the sample is determined as non-reactive, which indicates there is no detectable antibody in the sample. But if it's over or equal to 1, it's reactive, indicating that there are antibodies in the sample. So among these 86 samples, 80 samples were reactive in the total assay, and 6 were non-reactive, so giving a sensitivity or percent of positive agreement of 93. This is the overall agreement. And when we look to break down the time of sample collection in relationship to symptom onset, as you can see that samples collected within 8 days, among those 20 samples, 16 were positive, 4 were negative. So this gave you a sensitivity of 80%. But later we did additional tests using a neutralizing antibody assay and found that those 4 samples that show negative in this assay really didn't have any neutralizing activity. So we can either include this 4 sample or remove those 4 samples from the calculation. So if you include that, the sensitivity is 80%. If you remove that, it's 100%. Either one for sample collected within the first week of symptom onset, this is very sensitive. And for samples collected after 8 days, we show 100% positive, so gave us 100% sensitivity in PCR positive samples collected over 8 days after symptom onset. And among those 17 samples that collecting time is unknown, 15 were reactive and 2 were non-reactive. So we believe this is a very sensitive assay. And the specificity of the assay was evaluated using 400 negative samples that were collected before the pandemic. These are from normal, healthy blood donors. And all 400 gave non-reactive results, giving us 100% clinical specificity in this panel of samples. So one question is whether this assay shows cross-reactivity with common coronaviruses, those coronaviruses that induce common cold and are likely to be around through the years, and a lot of the people get exposed to those viruses. We did not do any direct tests because we do not have samples that have antibodies. No known samples have antibodies to those coronaviruses. However, through the publication, we know that over half of the healthy population has antibody to the common coronaviruses. So, which is very likely that among those 400 healthy blood donors, there are samples that contain antibodies to common cold coronaviruses. And the fact that 100% of this sample, 400 samples, show non-reactive to this assay highly suggests that the assay does not cross-react with antibodies to common cold coronaviruses. So again, this is not a direct testing, but a very logical indirect indication that the assay doesn't have cross-activity with common cold coronaviruses. We also use samples, interference samples that have potential cross-reactivity. These are the samples with antibodies to other respiratory viruses, as well as the HCV antibody and anti-nuclear antibodies. And all the sample gave us negative results. Again, this indicates 100% specificity. When we developed the assay, our R&D team not only focused on the sensitivity, but also focused on high specificity. The reason being that high specificity, only a very high specificity can give you confidence about a positive result. So as we all know, the certainty of a positive result is impacted by the prevalence of the disease. Even though we do not know for sure the true prevalence of SARS-CoV-2, but based on the current publication, that it's probably in most of the areas is single digit. So the lower the prevalence, the higher requirement of specificity. Here I gave you a prevalence of 1 just for the sake of an example. A specificity, if the prevalence is 1%, assay with a specificity of 99.9 gives you a positive predictive value of 90%. So that's pretty high. Tells you that if you have a positive result, most likely you have antibodies to the targeted viruses. But another assay has a specificity of 99%, which is also high when you look at the number. At 1% prevalence only gives positive predictive value of 50%. Basically, it's a 50-50 chance, like flip a coin, right? That result is not very reassuring. So we need to have a highly specific assay so that gave you the certainty of the positive result. So how can this assay be used and potentially be used, right? Because we know that the total assay is a very high sensitive and specific assay, really it can be applied to a much broader clinical utility. And as we discussed, because this assay is sensitive, it detects all types of antibodies. It may be used as an aid in diagnosis for acute or recent infection in conjunction with the PCR result, especially at a time that when the PCR result is not very sensitive. PCR tests normally are sensitive by technology. However, because the sample is coming from nasal swab, sometimes the sensitivity of PCR can be compromised due to the sample collection, the pre-analytical errors, or due to the disease progression, because through the disease progression, the virus migrated more to the lower respiratory tract, so it may not be detected at the upper respiratory tract. So this is where the serological test or antibody test that has the sensitivity can play a role to increase the accuracies or the sensitivity of detection of the viral infection. And of course, it can also be used for epidemiological studies. So this is to evaluate the magnitude of the COVID-2 infection in a given community by assessing serum prevalence, so to see how many people actually showing antibody positive indicate that these people have been exposed to the virus, whether they have symptoms or not, either currently or previously. And this also can show, give you a hint of the control of the infection spread or infection control by detecting the trend of seroprevalence. So whether this seropositive population is increasing or decreasing or maintained, this tells you how well you control the spread of the spread of the infection. And also this can be used for convalescent plasma donor identification, so to tell who has the antibodies that potentially can be qualified as a donor to the donor of their plasma for the treatment of severe COVID-19 patient. So again, very high sensitivity assay with a very high sensitivity and specificity with a very good clinical performance can pretty much fulfill the need of a broad clinical utility need. So this is a total test. Then let's take a look at the IgG test, the VITROS IgG assay. And this is also a qualitative assay that only detects the IgG antibody. Currently can be used in serum, and our R&D team is still working on the plasma. So hopefully we will have the plasma claim very soon. And the claim they intended to use is aiding identify patients who have adaptive immune response to SARS-CoV-2. So the presence of IgG indicating recent or prior infection. So when you look at the structure of the assay, this is again the VITROS micro-well technology. The well is coated with the biotinylated recombinant S1 protein, the same antigen as we used for total assay. And the assay will capture any antibodies that can react to the S1 protein, S1 antigen, and was detected with the murine anti-human IgG monoclonal antibody. So only the IgG being captured by the S1 protein will be detected in this assay. So this only detects the IgG antibodies. And as you can see, because we use S1 antigen, this assay can also detect the neutralizing IgG antibodies to S1 protein. Again, the assay is not sensitive to biotin interference. So the sensitivity of this assay has been evaluated with 58 samples. Again, this is collected from 58 individuals. patients, one sample, one patient. These samples are PCR positive for COVID-2. And among those 58 samples, 18 of them were collected before 15 days, and 40 of them collected after 15 days. The same result presentation and interpretation as the total assay. So for the 58 samples, 51 show reactive and 7 show non-reactive. If we break the results by time of collection in relationship to symptom onset, we can see that samples collected before 15 days show a sensitivity of 83.3%, and the samples collected after 15 days show a 90%. So this is still considered to be a sensitive assay, but when you compare to the total assay is more sensitive just because it detects all antibodies. So there might be IgA or IgM coming before IgG in some of the samples. And again, the specificity of the assay has been evaluated using 407 negative samples collected before the pandemics, and all 407 gave a non-reactive result, which gave the assay 100% specificity in the sample panel. And again, the same logic applies to this assay, that we do not believe the IgG assay has cross-reactivity to antibodies to other common coronaviruses. We included more samples, cross-reactivity samples, to evaluate the specificity. Again, all the samples containing antibodies to other respiratory viruses, also with the HCV and the anti-nuclear antibodies, gave a negative result, again supporting the 100% specificity. Same logic why specificity is important, that in a low prevalence situation, that small differences in specificity can give you a big difference in terms of the certainty of a positive result. So we really put a lot of effort to make sure that our assay is not only sensitive but also specific, so show high sensitivity and the specificity. So oftentimes people ask, how do you use this two assay? When do you use which assay, right? Again, because the total assay is the more sensitive one between these two. So really for aiding diagnosis, you want to have a very sensitive assay that can detect the early serial conversion. So we think the total assay is more suitable for using the aiding diagnosis, and also to manage a patient to detect the serial conversion during the disease progression, especially for hospitalized patients. This may give more information about the patient to ensure better patient management. But in terms of other utilities, both assays can be used interchangeably, because that for tracking the surveillance, that or for epidemiology studies, for understand the scope of the viral infection, or to understand the control of the pandemic, both assays can be used. And for convalescent plasma therapy, to identify convalescent plasma donor, both assays can be used. So it really depends on what's your needs and what do you want to use this assay for, so you can choose these assays according to your need, according to your patient's needs.